Fantastic. So today's webinar is our Microbiome Foundation's webinar presentation on dysbiosis and the gut organ axis with our Microbiome Labs clinical team expert, Sarah Ellis. Today, I will be dropping the link for the slides into the chat. So if you'd like to follow along, you can follow that link. If you haven't signed up for Frontify yet, go ahead and follow the prompt on your screen, and I will approve the request as they come in. And as always, you will receive a follow-up email with the instructions on how to access this webinar and slides, as well as all of our past webinars as well. So since everyone's time is valuable, we're going to get started today. So Sarah, if you'd like to get going.
Absolutely. All right, let me share my slides. And then I'm going to full screen slideshow. Maybe. There we go. All right, everyone, if you could put a 1 in the chat, if you can see my screen well, and if you can hear me well, that would be great. And then as soon as I see a couple of 1s, we'll get started. All right, there we are. Perfect. Hi, everyone. Thank you, Nikki, for that great introduction. I'm Dr. Sarah Ellis, and thank you for joining us for our second installment of the Microbiome Foundation's webinar series. Today, we are going to explore the systemic impact of gut dysbiosis. Many times when we discuss gut dysbiosis, it is centered around gastrointestinal symptoms and gastrointestinal disorders. But what we're knowing and learning through research is that gut dysbiosis can impact nearly every single organ in our body. And so today we are going to spend some time, let me get my cursor going. We're going to spend some time going over the brief overview of gut dysbiosis, the key differences between a healthy and leaky gut, and then we'll go through the impact that gut dysbiosis has on our overall health. So we'll discuss the gut-brain axis, the gut-heart, gut-lung, gut endocrine, gut immune, and gut genitourinary. And of course, at the end, we'll have a nice summary and we'll have time for a couple of questions as well. So let's get started. I'm going to be sipping some water in the background, so excuse me if there's a pause. All right, so let's do an overview of gut dysbiosis. And I also need to put all my devices on silent, so excuse me. All right, so dysbiosis can be defined as a reduction in microbial diversity and is often a combination of both the loss of beneficial bacteria as well as an increase in pathogenic bacteria. If we look at this image over here, let's see if I can get my laser pointer. Perfect. If we look at this top image in green, this is what the microbiome should look like in an ideal situation, what homeostasis or balance looks like. So we have a nice balance of all of these different types of bacteria. But dysbiosis can show up in a couple of different ways. First, we could have a pathobiont expansion. So the diversity is still there. There's still a decent amount of different types of bacteria, but the pathogens in red and orange have begun to kind of take over the microbiome. Dysbiosis could also look like reduced diversity. So instead of having all of these different types of bacteria, patients after going through antibiotics oftentimes are left with only a few different types of bacteria in the microbiome. And then, of course, dysbiosis could look like the loss of beneficial microbes. So while the pathogens are within range and have a good balance with each other, you can see here that we've lost quite a bit of the beneficial bacteria. Now, when you're looking on a stool test, there are a number of signs that can point out gut dysbiosis. There's often going to be decreased diversity, both alpha and beta diversity, so both the number of species present in the gut and the quality and balance of good to bad bacteria in the gut as well. We'll also see a decrease in the levels of protective keystone species that have a unique impact, on the gut microbiome. And then we may also see a decreased production in healthy byproducts like short-chain fatty acids. Now, dysbiosis can also show signs of increased negative impacts or products of the microbiome. So we can see increased pathogens, increased production of harmful byproducts like histamine and LPS, and hydrogen sulfide gas. And then, of course, increased inflammation as well. Okay, so what puts a patient at risk of developing gut dysbiosis? Well, there are a number of dysbiosis risk factors. These are some of the most common. And as we go through this list, I want you to think of how often your general patient base is exposed to these risk factors, because the frequency and severity of exposure can also correlate with the severity of dysbiosis as well. So risk factors include prescription antibiotics, natural antimicrobials, pesticides, infections, excessive alcohol use or smoking, stress, poor sleep, intense exercise, and also a poor diet that usually looks like the standard American diet that is rich in saturated fats. Now, let's compare a healthy to a leaky microbiome. So on the left here, we have a healthy microbiome that has low levels of pathogens, a strong mucosal layer, low mucosal inflammation, strong tight junctions between these colon cells here. But when we look at a dysbiotic gut, we can see here that it first starts with an overabundance of both mucus degrading and toxin producing pathogens. So we're seeing this molecule here, LPS, which is present on the surfaces of gram-negative bacteria. And so when a lot of LPS is present in the gut, we're going to see weakened tight junctions. So you can see instead of the colonocytes being very close to each other, we have, weakened tight junctions, inflamed intestinal cells, and as that lipopolysaccharide toxin makes its way through the weakened tight junctions and inflamed cells, we're going to see an increase in inflammation. Again, when looking at a stool test, there's a number of things present that can point to dysbiosis. And we can see the impact that being exposed to these risk factors has on the microbiome. So as we can see, the alpha diversity for this patient is quite low. So that's one of the first signs of dysbiosis. This patient only has 98 species in their microbiome, and we really want to see this number closer to 200. We can also see that there is an imbalance in the 4 major groups of bacteria in the microbiome. Particularly, there's quite a bit of the inflammatory group of bacteria, proteobacteria, which is another sign of dysbiosis as well. Now, we've talked a lot about the impact that gut dysbiosis can have on inflammation, but really that begins with understanding that the bacterial contributions of a healthy gut can look very different from the bacterial contributions of a dysbiotic gut. So a gut microbiome that is rich in commensals will make short-chain fatty acids, which is great for our colon health, supports good immune health, and many others. Healthy commensals will make vitamins, antioxidants, neurotransmitters, and will also optimize the gut pH, because a healthy gut pH will support the growth of commensals versus a more elevated pH can support the growth of pathogens. Now, if you have a microbiome that has quite a bit of pathogens present, you may find that the gut is making ammonia, hydrogen sulfide, methane gas, other toxins and virulence factors, as well as LPS. So gut dysbiosis, as we'll talk about during our time today, it impacts so many different conditions. It can be involved in hypothyroidism, a number of musculoskeletal conditions, of course, gut issues as well, including IBD and IBS. It impacts our metabolic health as well, involving obesity and type 2 diabetes. We'll talk more about how it impacts cardiovascular disease and as well as the gut and the brain. We see here on this slide the impact of dysbiosis on the human microbiota, and it ranges from skin issues to atherosclerosis, psychiatric disorders, lung diseases, as well as metabolic conditions as well. All right, so let's get those notepads out, get ready to take some notes. We're going to go from the top down, and we're going to start with the brain. So let's talk about dysbiosis and the gut-brain axis. All right, so our gut is powered by its own nervous system, the enteric nervous system. And there are a lot of neurons in the ENS. There are more neurons in the gut, over 100 million, than there are in the entire spinal cord. And this entire nervous system is accessible to the microbiome. And that power of the enteric nervous system helps create and facilitate the gut-brain axis, which is a bidirectional communication pathway. This image shows that so many factors with the host, including diet, exercise, mood, stress, and even their gender, can impact not only the type of hormones and neurotransmitters that are produced and released, but those hormones and neurotransmitters can also impact the gut microbiome. So, for example, norepinephrine, epinephrine, dopamine, estrogen, and progesterone, all in different ways, can impact bacterial growth in the microbiome. These hormones and neurotransmitters have been shown to impact biofilm growth, the level of resistance that the bacteria has to the host, as well as their virulence. Now, one of the main ways that the gut communicates with the brain is the vagus nerve. Now, this bidirectional communication pathway and really the whole gut-brain axis involves a variety of different organ systems. So, of course, it involves the nervous system, so the vagus nerve, the brain, and the spinal cord. But it also involves the endocrine system, and specifically the HPA axis, or hypothalamus, pituitary and adrenal gland axis. It also involves the immune system and the gut microbiome. Now, we've talked about the ENS and its the role that it plays and its impact on the gut-brain axis. But it's important to note that the hormones and peptides that are released by the enteric nervous system can cross the blood-brain barrier, much like ghrelin, for example, or a hunger hormone. And those hormones and peptides can act with the vagus nerve. Now, when we're thinking about the gut and the brain and the way that they communicate, it's important to know that they don't communicate equally. So it's not like the gut is talking for 50% of the time and the brain is communicating for 50% of the time. In fact, they're a lot like two best friends. One is an introvert, the other is an extrovert. The gut is the extroverted friend. It is a chatty patty. It's telling the brain so much about what's going on. You know, we ate this, we responded to this, the microbiome is making these things. Whereas the brain is really that introverted friend. They're listening, they're taking in all of your information. And when they do communicate with you, they're saying something impactful and powerful. And so the relationship between the gut and the brain are very similar. And that can definitely, it's one of the ways that we can figure out whether some of the patient's issues is the gut ability to talk to the brain or the brain's ability to communicate with the gut. All right, the vagus nerve, a little bit of details about the vagus nerve. It is our 10th cranial nerve, and it is the fastest and most direct route that connects the gut and the brain. It innervates a number of visceral organs. And so it innervates the neck, so the pharynx and the larynx that's involved with swallowing. With the chest and the thorax, it is involved in the parasympathetic supply to the heart. So the vagus nerve and vagal tone can actually help reduce the heart rate. Of course, it is involved in the intestines and helps regulate the contraction of smooth muscles and glandular secretions. And it also has afferent receptors in the esophagus, stomach, small intestine, and even some sensory endings in the liver and pancreas as well. Now, the vagus nerve is responsible for a number of functions in the body. It regulates internal organ functions, primarily the ones that are autonomic. So, you know, our digestion, our heart rate, our respiratory rate. but it also is responsible for a lot of vasomotor activity and certain reflex actions. Coughing, sneezing, swallowing, and vomiting are all of those that are impacted by the vagus nerve as well. Now, all of this is important. I laid that background out because we are noticing, and I'm sure you as an integrative and functional medicine provider, you are noticing a rise in what's called the functional gastrointestinal disorders, or FGIDs. Now, these are a unique group of disorders that are characterized by a common group of symptoms. So usually patients that present with these FGIDs, like functional dysphagia, functional heartburn, rumination syndrome, and IBS, they present with kind of the similar constellation of symptoms: abdominal pain, dysphagia, dyspepsia, constipation, and bloating. Researchers have found that these conditions begin to develop due to alterations in the gut-brain communication. And in fact, the number of patients that are presenting with these symptoms and conditions are increasing, and they actually account for over 30% of outpatient gastroenterology visits. And not only that, they are also accompanied by a lot of other functional conditions like chronic pain, chronic fatigue syndrome, and other functional syndromes like those. All right, well, it wouldn't be a MBL webinar without a pop quiz. So which of the following is considered to be a cause of gut-brain axis dysfunction? Let's see. Is it LPS endotoxemia, fasting, candida infections, or none of the above? So let's hop in the chat and let me know what answer you're putting in for this question. A, B, C, or D? I'll give you guys a few seconds. All right, lots of A's. You guys are on it. I love it. Let's see if we can get this going. That's right. It is lipopolysaccharide endotoxemia. And so the next piece of our presentation, we're going to talk about LPS endotoxemia and why it is a common cause of gut-brain axis dysfunction. So LPS-driven inflammation is because of or caused by the lipopolysaccharide endotoxin, which is present on the surface of gram-negative bacteria. Gram-negative bacteria actually make 50 to 60% of our bacteria in the microbiome. So over half of the bacteria in our microbiome are gram-negative, which means that over half of the bacteria in our gut have lipopolysaccharide. Now, when it's inside of an intact, healthy bacterial cell, LPS plays no issue for us. There's no concern with that. However, when the bacteria die, they lyse open and they release LPS. And when LPS is free in the microbiome, it can cause, as we saw earlier in that schematic, it can cause weakening of the tight junctions, inflammation, and eventually will be able to seep out into circulation. And the first place it goes to is the brain. And in the brain, it can wreak havoc. So this study is one of the studies that we mention a lot, but this is one of the first few studies back in 2008 that showed that the gut-brain barrier, as well as the presence of lipopolysaccharide from the gut, plays a role in the inflammatory presentation and pathophysiology of depression. So let's see, that was about 16 years ago. So 16 years ago, researchers were seeing that lipopolysaccharide, when it is able to come out of the gut and enter circulation, it plays a significant role not only in the presentation of depression, but also the severity of depressive symptoms. When we looked at, or researchers, when they looked at the gut microbiome of patients with anxiety and depression, they saw some interesting things. What they found is that patients with anxiety and depression had far fewer bacteria that produced short-chain fatty acids. So they had a reduction in keystone species like Faecalibacterium. But they also saw an increased presence in inflammatory bacteria. So they had more enterobacteria and desulfovibrio, which is a group and family of bacteria that produce hydrogen sulfide gas. And so these patients were more likely to experience dysbiosis. And we can also see that some confounding factors that may play a role in this presentation of the microbiome is, of course, diet, whether or not they're using and taking psychiatric medication, whether they meet certain clinical stratification criteria, as well as their gender and sex. All right. This study found that microbiome-derived lipopolysaccharide was found in the perinuclear region of the Alzheimer's disease brain. So those beta amyloid plaques have been found to be quite attractive for lipopolysaccharide molecules. And so researchers have found within those beta amyloid plaques, microbiome-derived lipopolysaccharide molecules, which confirms that LPS can cross the blood-brain barrier. And as we know and as we've seen, LPS can cause inflammation, and Alzheimer's disease is associated with neuroinflammation. Not only does LPS or is LPS implicated in the progression and severity of anxiety and depression, as well as Alzheimer's disease, it has also been indicated in Parkinson's disease. Basically, what researchers have found is that the inflammatory response associated with the presence of lipopolysaccharide in the brain can also increase the alpha-synuclein aggregations that are present in the brains of patients with Parkinson's. And this induction of those aggregations is started by the translocation of lipopolysaccharide into the brain via the vagus nerve. So let's talk about what happens in the brain when lipopolysaccharide when the brain is exposed to lipopolysaccharide and that inflammatory process begins. So of course, in the gut, we have LPS that's on the surface of gram-negative bacteria. Eventually, it does translocate across a leaky gut and enter circulation, where it upregulates the immune cells, particularly the production of pro-inflammatory cytokines like interferon gamma. Now, LPS also will increase the enzyme IDO. Now, IDO is very important because this is an enzyme that impacts how our brain uses tryptophan. And we'll talk about this in a couple of slides as well. But this is an enzyme that can determine whether or not tryptophan goes down a neurotoxic pathway or a neuroprotective pathway. Now, the increase in expression of this IDO enzyme means that less and less tryptophan is going to be available to make melatonin and serotonin, and instead it is going to be shuttled into that toxic pathway. Now, here's the schematic that I wanted to share that best explains how tryptophan metabolism is impacted by inflammation. So tryptophan can be received in our body through diet, or our microbiome can make tryptophan as well. Now, the microbiome may not make tryptophan or as much tryptophan. The inflammation present in the gut can reduce how much tryptophan is available to be used in this neuroprotective pathway. So for example, if you have a patient with a lot of inflammation and a lot of stress, that is going to upregulate that IDO enzyme. And when that IDO enzyme is upregulated, it is going to shuttle tryptophan down this neurotoxic pathway that ends up making quinolinic acid. When you have a patient that has most of his tryptophan going down this pathway, they usually present with a depressed mood, irritability, poor resistance to stress, poor sleep, and poor concentration. But if we're able to reduce this inflammation and support them with a healthy stress management tools, then that frees up more tryptophan to be used to make serotonin for a balanced mood and melatonin for better sleep. When we're able to shuttle tryptophan into this neuroprotective pathway, we're able to increase our patient's sleep quality. They have a better circadian rhythm. A better circadian rhythm. They have a positive outlook on life, more resilient to stress, and they have a better memory. All right, so that was the gut and the brain. Now let's talk about the gut-heart axis. So dysbiosis does play a critical role in the development of chronic disease, especially cardiovascular disease. And there have been several studies to investigate the crosstalk between nutrition and the microbiome, intestinal permeability, and cardiovascular disease. And what they found is that there are several microbial metabolites that have been identified as having an impact on the development of cardiovascular disease. Some of those metabolites include TMA, which we'll talk about in just a few seconds, LPS, bile acids, serotonin, and short-chain fatty acids as well. All right, TMAO, trimethylamine N oxide. TMA is produced in the gut from sources of lecithin, choline, and L-carnitine. So seafood, dairy, and eggs, as well as meat, produces TMA. Now, TMAO is produced when the TMA that's produced by our bacteria reaches the liver, and the liver produces TMAO. Now, TMAO is the issue because this is a biomarker that has been shown to be pro-atherogenic. So it promotes the development of atherosclerosis. And it's been positively correlated with early atherosclerosis. TMAO has also been implicated in type 2 diabetes, metabolic syndrome, heart attack, of course, atherosclerosis, and also chronic kidney disease. In a meta-analysis of over 19 studies, it showed that the higher that the plasma TMAO levels are, it's been linked to an increased risk in cardiovascular events like stroke, MI, and death. Now, the higher the microbial diversity, the lower the amounts of TMAO that is produced in the body. So let's look at some research studies. In 2017, researchers compared the stool samples and stool microbial profile of patients with atherosclerotic cardiovascular disease and healthy adults. And what they found is that the gut microbiome of patients with atherosclerosis showed an overabundant levels of pathogens like Enterobacteria, Klebsiella, and Streptococcus. It also saw a depletion in butyrate-producing bacteria, so those keystones, like Faecalibacterium. There was also a presence of inflammatory metabolites and toxins, like TMA and TMAO, and also a reduction in the fermentation of dietary fiber and the production of short-chain fatty acids. Now, what's interesting here is this article shared that when they looked at the atherosclerotic plaques in the vasculature, they were actually able to find bacterial DNA inside of those atherosclerotic plaques. So this is a further confirmation that the translocation of lipopolysaccharide from pathogens in the microbiome has been found not just in the beta amyloid plaques in the Alzheimer's brain, but also the atherosclerotic plaques in the vasculature. Now, this same study highlighted not just the type of cardiovascular disease, but the changes in the gut microbiota that's often seen in those diseases and the most common metabolites associated with those diseases. So with coronary atherosclerosis, researchers saw an increase in these pathogens, and TMAO along with bile acids and lipopolysaccharide were found to be involved in the development of atherosclerosis. Now, with hypertension, we saw an increase in some of these species, but what's interesting is that The metabolites that had the most impact on hypertension was short-chain fatty acids, particularly propionate. And that's because these short-chain fatty acids, they actually adjust our immune cells, our T helper cells. And so short-chain fatty acids like propionate are able to adjust T helper 17 cells and then lower blood pressure. So these short-chain fatty acids are not necessarily associated with an increase of hypertension, but rather a decrease. And then some of these species of bacteria and their levels have been associated with heart failure as well. All right, so let's talk about dysbiosis and coronary artery disease. So previously we talked about general atherosclerosis, but right now we're going to talk about coronary artery disease, or the disease in the coronary arteries that provide blood to the actual heart. So this 2019 study looked at the microbial composition of the gut of patients with CAD. And what they found is that the microbial composition, as well as the metabolites that are made in the gut, like TMAO, changed significantly as coronary artery disease progressed in severity. So the gut microbiome of patients with coronary artery disease showed some of the following patterns. Much like atherosclerosis, there was an increase in proteobacteria or inflammatory bacteria like Klebsiella and Streptococcus. There was also an increased production of toxic metabolites like TMAO and LPS and hydrogen sulfide gas. But there was also a decrease in short-chain fatty acid production, particularly low butyrate. Now, in hypertension, dysbiosis can also play a role in the development of hypertension as well. It's one of the most common risk factors associated with cardiovascular disease. And in fact, the gut microbiome has been shown to play a role in blood pressure, which we talked about a couple of slides ago. And so the researchers looked at the stool samples of patients with hypertension and found that those patients tend to present with reduced alpha and beta diversity. So they have a lower number of bacteria in their gut, and the quality is also reduced as well. There's an increase in the genus Prevotella, as well as imbalances in the number of protective species like Bifidobacterium and Lactobacillus versus the presence of more inflammatory bacteria like Streptococcus and E. coli. Now, this is very important because these species of bacteria can make neurotransmitters. And we know that neurotransmitters can impact the autonomic nervous system, vascular tone, and eventually blood pressure and heart rate. Elevated TMAO is positively associated with hypertension, as well as reduced short-chain fatty acid production. Now, this study was really, really interesting because it showed that a fecal microbiota transplantation from hypertensive patients to germ-free mice, so these are mice that have not been inoculated with any bacteria, they do not have any bacteria in their bodies, they received a fecal transplantation and began to develop high blood pressure and saw a subsequent increase in the blood pressure of those mice, which shows the impact that the stool and the gut microbiome and the metabolites made by the bacteria has on our heart rate and blood pressure. All right, let's keep going and talk about the lungs and how gut dysbiosis can impact our lungs. All right, let's talk about gut dysbiosis and asthma. So dysbiosis and subsequent dysregulation of certain immunological processes in the gut can affect the onset of the disease of asthma, its clinical characteristics or how patients present with asthma, as well as how well they respond to treatment. Now, it's important to know that asthma is considered an atopic disease, and atopic diseases are directly linked to the presence of gut dysbiosis as well as leaky gut. And so it's very difficult to talk about atopic diseases like asthma and eczema without also discussing the gut and the gut microbiome. So this study or this image connects the symptoms and clinical manifestations with the gut microbiome. And so not only are there different phenotypes of asthma, exercise-induced, early onset or late onset, but there's also endotypes, which means that different types of asthma can present differently with the gut microbiome or can cause a different type of microbial composition in the gut. So if you have a patient with type 2 asthma, they may be more likely to have a microbiome that has increased numbers of pathogenic bacteria like Haemophilus, Klebsiella, and Streptococcus, as well as decreased levels of potentially beneficial or anti-inflammatory species like Actinobacteria, which is our bifido species here. Non-type 2 asthma, you may see an increase, again, in pathogenic bacteria like Fusobacterium or Porphyromonas. And then you have decreased levels of different families, including the Lactobacilliaceae family. I really just wanted to focus on asthma for the lungs for the sake of time, but gut dysbiosis has been shown to play a role in COPD as well. All right, so let's talk about the endocrine axis and our hormones. And we're going to start with diabetes. So the gut microbiome, of course, plays a lot of roles, but it does help regulate our hunger and satiety by releasing different hormones. It could also play a role in insulin resistance and insulin sensitivity. So there's been a number of studies that have looked at the gut microbiome, the bacteria that are present in the gut in patients who have type 2 diabetes. And what we have found is that there are specific types of bacteria that have been shown to support glucose metabolism and insulin sensitivity. And in a couple of slides, we'll talk about the differences in the gut microbiome from type 2 diabetes patients compared to healthy patients. But what we know is that several lactobacillus species, like Lactobacillus fermentum, Lactobacillus plantarum, and Lactobacillus casei, along with Roseburia, Akkermansia, and Bacteroides fragilis, all of them play a role in our ability to support healthy blood sugar regulation. Now, what's interesting is that metformin, which is the first-line pharmacotherapy for type 2 diabetes, has been shown to act primarily in the gut. So it does not act on the pancreas. Instead, it changes the landscape of the microbiome to help facilitate reduction of blood sugar. So it increases butyrate production, it increases propionate production, and has been found to increase levels of Akkermansia muciniphila. Again, metformin, its mechanism of action acts on the gut, and it has been found to impact and alter the microbial composition. So at the phylum level, when we're looking at kind of the more broad combination or groups of bacteria, we have found that patients with type 2 diabetes have a lower abundance of firmicutes or firmicutes, depending on who you're talking to, than the control group. Now, this is important because if any of you have ever run a BiomeFX stool test or had a consultation with myself or some of the other consultants, you know that we talk about dysbiosis ratios and we compare the 4 major groups of bacteria in the gut. And firmicutes is one of those 4 major groups of bacteria, but it is also the group that has the most of those protective and special keystone strains. So the lower amount of firmicutes that you have, usually the lower levels of keystones you also have. Type 2 diabetes patients also had elevated levels of proteobacteria. They had fewer levels of Roseburia, which makes butyrate, and as we talked about in the previous slide, plays a role in blood sugar regulation. And also type 2 diabetes often, those patients often had low alpha diversity as well. Now let's talk about thyroid. When preparing for this lecture, I really enjoyed researching the connection between the gut dysbiosis and thyroid because it plays a big role in our ability to detoxify lipopolysaccharide. So just as a overview, the thyroid is a target, is an organ that is involved in our metabolism. And in all honesty, it's involved in so many different functions in our body, from our heart rate and our breathing, the way that we metabolize glucose, our menstrual cycles, and so much more. The gut is a target organ for our thyroid hormones. So thyroxine, or T4, and triiodothyronine, T3, can have an impact on the gut. In fact, thyroid hormone receptors are expressed on the surface of our intestinal epithelial cells, and disruption in the microbial composition has been associated with Hashimoto's thyroiditis, Graves' disease, and Graves' orbitopathy. Now let's talk about the impact that thyroid disease has on lipopolysaccharide toxicity. And that would start with a discussion about intestinal alkaline phosphatase, or IAP. This is an enzyme, a brush border enzyme, that is primarily produced and expressed in the duodenum, in the small intestine. Now, IAP works to detoxify LPS. It makes it less toxic, less inflammatory, and helps reduce that inflammatory burden associated with LPS. It also prevents it from being translocated into circulation. Now, what's interesting is that researchers have found that this IAP gene is T3 responsive. And so this links the expression of IAP, or intestinal alkaline phosphatase, to the levels of thyroid hormone. So what does this mean for your patients? If you have a patient with hypothyroidism, whether it's Hashimoto's thyroiditis or just non-autoimmune hypothyroidism, they're going to have lower levels of T3. And the lower levels of T3 are associated with low levels of intestinal alkaline phosphatase, which means that your patients with hypothyroidism are less able to detoxify LPS and more susceptible to LPS toxicity. In animal models, they found that a decrease in the IAP gene in hypothyroid rats, or hypothyroid rats, had a decrease in the expression of the IAP gene. And when they knocked out the IAP gene in zebrafish, they became more susceptible to LPS toxicity. So if you have a patient with hypothyroidism, you definitely want to make sure that you're supporting their gut health and checking in on those levels, either the levels of LPS or the presence of LPS containing bacteria. Now let's talk about those microbial patterns that are associated with autoimmune thyroiditis. What we see is that there are a number of bacterial strains that are associated with a decrease in the expression of antibodies against the thyroid tissue. So Bacteroides, the Dorea species, and one of our favorite keystones, Faecalibacterium, is associated with lower levels of these antibodies. Now, an increased expression in these thyroid antibodies include Blautia species, Lactobacillus species, Allostipes, and Ruminococcus. Now, patients with Hashimoto's thyroiditis were found to have more Lachnoclostridium, Bilophilo-Wadsworthia, and Klebsiella pneumoniae in their stool. Now, Bilophila and Klebsiella are both gram-negative. They contain LPS and are quite inflammatory. All right, this is a great transition into the immune system. So let's talk about the gut microbiome and the immune axis. Of course, we know that the gut microbiome plays a significant role in our immune system and our immune health. Most of our immune cells are present in the gut, and a healthy microbiome is incredibly necessary from early life because our gut bacteria helps train our young immune system. So having a healthy, balanced microbiota can help train and teach the immune cells which compounds and antigens are friends or foe. And so when a child or a baby is born to a mom with a dysbiotic gut, or they're born through C-section and aren't able to be exposed to mom's vaginal microbiota, they are more likely to have dysbiosis, and they're more likely to have atopic diseases, which is a reflection of poor immune tolerance. So their immune system didn't really get a chance to get trained by a strong microbiome. And so they're more likely to kind of be confused as to whether or not something is a friend or a foe, which means they're more likely to develop autoimmune conditions. Now, gut commensal bacteria have a very vital roles in the establishment of a regular innate immune system. And in turn, the dysbiosis of the gut microbiome can cause an alteration in our innate immune system or our first line of defense, and of course, vice versa. Microbiota colonization in early life, as I mentioned, does affect the development of our T cells, and it determines how those T cells are differentiated. Do they turn into Th1, Th2, and Th17 cells that are more inflammatory? They produce more inflammatory cytokines to help fight pathogens. Or are we able to differentiate into regulatory T cells, which are kind of like our fire extinguishers? They cool off the inflammation caused by Th1, Th2, and Th17 and kind of bring everything back to homeostasis. If the microbiota of an infant, oh, there we go. If the microbiota of an infant is not properly developed, you're more likely to have an imbalance in the levels of differentiation between these T cells. Oh, I still have the audio on this slide. So let's talk about the genitourinary axis. All right, so let's start with the vaginal microbiome. Unlike the gut microbiome, we do not want high diversity in the vaginal microbiome. With the gut, we like to see lots of bacteria and lots of different bacteria. But in the vagina, we really only want to see lactobacillus species. A healthy vaginal microbiome has a more acidic pH between 3.8 and 4.5. And that's thanks to those lactobacillus species that produce lactic acid and keep the vaginal pH nice and low. Now, there are 4 dominant lactobacillus species: Lactobacillus crispatus, Lactobacillus gasseri, Jensenii, and Einers. Now, it's important to note that Einers can go both ways. It can be associated with dysbiosis or a healthy vaginal microbiome. Now, lactobacillus species are critical for a healthy vaginal microbiome. As I mentioned, they do produce lactic acid, which helps reduce the pH, but lactic acid, bacteriocins, and hydrogen peroxide, all of those are produced by lactobacillus species. They can also help control the pathogen abundance as well. So lactobacillus creates a lot of different compounds to keep pathogens at bay. It can also inhibit the adherence of pathogens. So it prevents pathogens from adhering to the vaginal mucosa by making surfactants that compete with attachment sites. So it kind of blocks off, if you think of it like a key, it kind of blocks off that key so that the pathogen can't get locked into the mucosa and set up shop. All right, the vaginal microbiome can be impacted by similar dysbiosis risk factors associated with the gut. So, of course, age and hormones play a role in the vaginal microbiome. Sexual habits, medications, the use of different vaginal products, diet, stress, tobacco use, and of course, the gut microbiome. Now, I mentioned the gut microbiome because there is a gut-vaginal microbiome crosstalk, mostly due to the proximity of the anus, the vaginal opening, and the urethra. Now, being in close proximity does mean that there's a potential for the translocation of species present in the colon to make their way into the vaginal canal as well as the urethra. And so this can play a role in the development of bacterial vaginosis and UTIs. Now, women with BV tend to have a lot of that BV-associated bacteria in their gut. So you may not be surprised to see Gardnerella vaginalis and Prevotella bivia and even mycoplasma present in their stool sample. Now, gut probiotics that contain lactic acid producing bacteria like lactobacillus and the bacillus spores, they can help maintain a healthy vaginal pH and block the growth of pathogens. Now, women with higher levels of Lactobacillus crispatus in their stool were more likely to have a healthy vaginal microbial composition. So the ingestion of oral vaginal probiotics can impact and improve vaginal health by introducing and sustaining Lactobacillus species into the gut. Now let's talk about the bladder. The bladder does contain bacteria, and it actually has a basic baseline of healthy good bacteria. So a healthy bladder is lactobacillus dominant. Now, different conditions like UTIs, particularly recurrent UTIs, interstitial cystitis, and overactive bladder and urinary incontinence can present with different bacteria. So, for example, recurrent UTIs were more likely to see more E. coli, Gardnerella, and Pseudomonas present in the bladder. With interstitial cystitis, we're more likely to see Brevibacterium or Citrobacter, or even Lactobacillus gasseri as well. And then with overactive bladder and urinary incontinence, we can see Atopobium, Gardnerella, Proteus, and even Smithia as well. Now, the urinary microbiome can fluctuate. A healthy bladder has a pH of 4.6 to 7, and like I said, is mostly lactobacillus. But there are a number of risk factors that can lead to UTIs and chronic UTIs. Of course, indwelling devices like catheters and age. Young children, particularly young girls, are more prone to have UTIs because of being in diapers, as well as the elderly are more likely to have UTIs as well due to issues with urination, particularly men that have benign prostatic hyperplasia or BPH. Low estrogen, so women, postmenopausal women, are more likely to have UTIs and particularly recurrent UTIs. And then, of course, a number of other risk factors like barrier contraceptives, vaginal infections, and even spinal cord injuries, antibiotics, and diabetes, which can impact the presence of immune cells to help fight off infections. Like I said, the UTI bladder has a higher pH, about 7.8 to 9, and that facilitates the survival of E. coli, Enterococcus, Staphylococcus, and of course, a reduction in lactobacillus. All right, we made it through with just enough time for questions. In summary, dysbiosis is an imbalance between beneficial and harmful bacteria. This microbial imbalance impacts all areas of our health. And gut dysbiosis has been associated with anxiety, depression, I can't talk today. Words are hard today. Neurodegenerative conditions like Alzheimer's and Parkinson's disease, asthma, type 2 diabetes, cardiovascular disease, autoimmune diseases, and urinary tract infections as well. Restoring gut homeostasis is critical for supporting overall health and reducing chronic disease. Thank you so much for your attention today. All right, let's check out these questions. Thank you, Dr. Aisha. I'm so glad that you enjoyed it. All right, we have about 8 questions. Okay, so there are slides. I believe the link to slides have been shared. What is the optimal gut pH? How do you measure it and how do you optimize it? I don't have, oh, Valerie, we don't have the slide link. Nikki, do you think you can post that? Or I think it might be in Frontify as
Yeah,
well.
I can drop that again, no worries.
Awesome, thank you. Okay, so I don't have the gut pH range in my brain right now. However, you can measure the gut pH by choosing a stool sample or a stool test that can measure pH. They measure the pH of the stool. And you optimize it by having more beneficial bacteria compared to pathogenic bacteria. So remember the commensals like the lactobacillus and bifido and Faecalibacterium and Akkermansia, they prefer a more acidic environment, whereas the pathogens, like the ones that have LPS, prefer a more basic or alkaline environment. So by optimizing the gut pH, you want to improve the microbial diversity, so spore-based probiotics. And honestly, the Total Gut Restoration Kit would be a perfect place to start and begin to optimize the gut pH. I hope that helped. All right. Is gram-negative bacteria commonly found in the gut? Yes, 50 to 60% of the bacteria in our microbiome are gram-negative. And so again, LPS is a normal part of the structure of the bacteria, but when the bacteria dies and that toxin is released, that's when it becomes a problem for us as host. So it's more so about making sure that you don't have an overabundance of gram-negative bacteria so that when they die, they release a ton of LPS. And so that's involved with keeping the microbiome balanced. All right, we have the notes and slides. There are some ways to decrease TMAO. Oh, Valerie. Oh, okay, hold on. Sorry. The slides were, let's see if we can do like this. There we go. Okay, I just put the slides link in the chat as well. So let me know if you guys can see that, or put a 1 in the chat if you click the link and it works. You're welcome, Valerie. Okay, what are ways to decrease TMAO? Of course, diet. And really, it's going to be about making sure that they're not eating a lot of those foods that make TMA, but also that they don't have a lot of that negative bacteria or harmful bacteria that can turn the foods into TMA. Now, again, TMA is not an issue. It's the TMAO that's produced in the liver. So it's more like getting to the root of the issue, which is trying not to have as much TMA be made in the gut that can be turned into TMAO. All right. Is IAP different from alkaline phosphatase? Yes. So the alkaline phosphatase that is measured is more so a reflection of like the liver and liver health and also bone health as well. But the intestinal alkaline phosphatase is a specific enzyme made in the small intestine. So it is a different enzyme. We have slides. Should a binder be used when killing LPS in someone with Hashimoto's? Absolutely. Whenever we are targeting LPS, we usually recommend Mega IgG 2000. That is our serum bovine immunoglobulin product that helps bind LPS. It binds gram-negative bacteria that produce LPS, so I would absolutely use a binder. Valerie, the microbiome supplements that are best to improve gut health, the Total Gut Restoration. It has our probiotic spores, Megaspore Biotic, our prebiotic Mega Pre, and our mucosal support product, Mega Mucosa. So you get all 3 in one, and you can take your time going through each month. Great, I'm glad you're using it. Thank you, Trevor, for helping me out. Trevor Miller says that the Heidelberg test helps measure pH directly. And the pH of the gut is around 5.1. So thank you, Trevor. I appreciate that. All right. I have one question from April. I know we're at time, so if you have to hop off, thank you so much for joining me. I appreciate your attention. Last question from April. She had a metabolics test that showed low carnitine. So she's supplementing. How do I know if my gut is sufficient enough to not convert TMA to TMAO? Very great question. Usually when patients are given carnitine as a supplement, we do want to make sure that we're supporting the gut with either a probiotic, like a spore-based probiotic, and maybe even a binder like Mega IgG 2000, just so that we are supporting the microbiome, continuing to introduce good, healthy bacteria that can help facilitate healthy levels of TMA so that a lot of TMAO won't be made in the liver. All right, I think that was the last question. I hope you all have a wonderful Wednesday, a great hump day, and I will see you all next time.
Yes, thank you all for joining us. And as I stated in the chat, we will be sending out the replay of this webinar as well as the slides. So have a good day and we'll see you next time.
Take care, everyone. Bye-bye.